Okay, so my presentation is online. I will speak about Pharmacolibrary. Welcome to a second exotic use case of Modelica in chemical, physiology, and medical domain. I'm Tomáš Kulhánek, and I work at the Flemish Institute of Technology in Mol in Belgium. But I still have affiliation and First Faculty of Medicine and Charles University, and my colleagues also are from that university, from the small company Creative Connection, and my colleague Filip Ježek is now at the University of Michigan, and another my colleague Marek, who spoke about the chemical library. It's at Institute of Clinical and Experimental Medicine in Prague, in Czech Republic. So in Vito, I'm an environmental unit, and we are a small group focusing on digital precision health. And we have several projects. Some of them are focusing on using fancy technologies like artificial intelligence. Another one is focusing on precision medicine. And my colleagues are mainly working with data. And when I got with some of my researchers at this organization, they use data and they don't use the proper modeling, the proper equations. They just have a script in Python developed by somebody else. And for example, they are focusing on pharmacogenomics and in other projects. So it was motivation to create some formalization and to make it more simple for them and also for me to develop and to model pharmacology domain. So what is the pharmacology? I already mentioned the pharmacogenomics. It's how the genetics affects the drug and body behavior. But the pharmacology is, there are more domains. The first one is pharmacokinetic. It is what the body does to the drug. And pharmacodynamic, what the drug does to the body. And there are other subdomains, but these three is the main. It is the target of the Pharmacolibrary. So pharmacokinetics, it's the way how the body acts to the drug. So when you take a medicine, for example, you take it orally, but you can also take the medicine just by injection inside the venous system or inside the muscle. And then there is some distribution, usually using cardiovascular system, but also the other systems and metabolism, mainly in the liver. Some drugs are metabolized also in, for example, in lungs, and excretion. The majority of drugs are excreted in urine or in feces. And these processes can be expressed in mathematical equations. So we chose connectors. And there are two types of connectors. First, doesn't take care about the flow, meaning volume flow rate. And this first part of the pharmacology, it's called compartment-based modeling. And it's its simplification. So in this first part, we chose potential variable as mass concentration and flow variable as mass flow rate. This is also chosen in different libraries, these variables, but these quantities. But if we take into account the volumetric flow, we chose to have a potential variable pressure, flow variable volume flow rate, and stream variable, the mass concentration. So the mass concentration is in both connectors, but based on the what simplification of pharmacology we are modeling. So in the case of administration, the more simple administration is intravenous. You just put the drug inside your body and then it is distributed. So this is the first part, the first diagram. You can connect the variable dose component into standard Modelica components. Here is pulse, so you can distribute the drug periodically, for example, every 8 hours or every once a day. More complicated is enteral administration. So if you take a drug via mouth, and then there is some delay between it goes to the gastrointestinal system and starts to be absorbed. And so we have also there some lumen compartment. I will say about the mathematics later, and some first order kinetics is there as well. And distribution. In case of the compartment model, there is no flow rate, so the basic component here has the equation of the mass flow rate, derivative of the mass and concentration. And there is also the first order kinetics. The equation is there. But if we take into account the volume flow rate, the concentration needs to be mixed. And thanks to Modelica standard, the stream connectors is very helpful here. So we can calculate the mass concentration and mass flow rate using that equation, using the actual stream function. And if we go to metabolism, it is also possible to express the metabolism in Pharmacolibrary, but in roadmap I have to integrate it with the chemical library because it is much more powerful than this library in chemical reactions. And elimination is again first order elimination rate, usually described in pharmacology literature. So the most common model of a drug pharmacokinetic is one compartment model, and so there is a single dose which is distributed equally into some central compartment, but central compartment means that it is blood and probably some tissues, but almost ideally distributed. And based on the experiments, the drug doesn't react much more with other tissues. And for example, this is the one of the drug called gentamicin. It is antibiotic used in hospitals when there is some infection. And if you can also express this this model using the equivalent equation-based model. But what is interesting is that here is the simulation of the gentamicin during the 1 day, and the decline after administration is logarithmic. So in a logarithmic scale, it looks linear. And a lot of drugs behave this way, that there is a linear logarithmic decline, and like the therapeutic effort is to maintain the concentration of the drug above some level. Here, the MIC, it's a minimal inhibitory concentration. There are two compartment model. Some of the drug does not only distribute in blood plasma, but they also distribute to other tissues. So that can be expressed as a compartment, central one, including the bloodstream and the peripheral. Again, we don't know what the peripheral compartment, which type of tissues are there, but numerically it fits the data. And for example, midazolam is used for sedation and anesthesia. And in a normal scale, you see again something like logarithmic decline, but on the logarithmic scale, it's not linear. And it is because the peripheral compartment here works as a storage at the beginning of the simulation and also the distribution. The drug is dissolved in the peripheral compartment, and that it behaves like a source, the drug. So the decline flow is not so deep like on the first part of the simulation. And here you have also the data from the experiments. There are 3 compartment models that it is again more complex. Here is the example of fentanyl. Again, the drug is used for anesthesia, and for 10-hour simulation, you see here again something like logarithmic decline, but here see that the 2 peripheral compartments make this like inflection points. So, and some of the drugs are designed that the decline after some time after the administration is not so steep. So the effect of the drug is prolonged. Yeah. Fentanyl is also known that it is misused in some countries, and it has some social negative social impact, but that's not a part of this presentation. There are physiology-based models. The 1, 2, 3 compartment models are mainly based on the data. So the data fits the models, but here the physiology-based models takes into account, for example, the cardiac output, the lymphatic system flow, or detailed organ tissue behavior. So here in the physiology-based pharmacokinetic model, we have the compartment for all possible tissues, but it is very hard to measure the experiments in all the tissues. So it is also hard to get the parameters. But for some drugs, it was done, and here you have the absorption in good, then it goes to liver, and it is distributed through panels to lungs and then to other tissues. And for example, when you simulate it, only 10 minutes after some time, it is absorbed to the gut, but the most of the highest concentration is in the liver. And in the liver, they may become the most significant metabolic changes to the drug. So you see that in other tissues, the concentration is not going so high, but the liver can be also affected by side effects. So way some drugs are designed to not to harm liver or other tissues. In the library, we have so far modeled 5,496 drugs that are classified by the anatomical, therapeutical, and chemical classification. So it's all approved drugs, and some of the drugs are not used now because they were found that there are some toxics and other consequences, or better, some of the drugs they are more than once classified there. We'll show you some examples later. So at the first level, there are 14 groups. On the ATC level 3, there are another, I don't know, to 10 to 20 30 groups. And for example, paracetamol is in the nervous system, and maybe other analgesics and antipyretics. And in this group, there are, for example, the first one is acetylsalicylic acid, it's aspirin, and the paracetamol is the, I don't know, the 15, 16. So there is the one compartment model of paracetamol, simplified the parameters clearance and almost distribution is taken from literature, but we use the PubMed, drug bank, and also large language model to extract such information. So not all the drugs and information are correct, but most of them are now quite according to what this is published. So back to caffeine. Caffeine is also a registered drug, but it's in a cafe or tea, green tea or black tea. So how many of you drink coffee in the morning? Raise your hands. And how many of you drink black tea? Tea? Green tea? Somebody? Yeah. So majority coffee, and the caffeine is registered as the nervous system and but it can be also used in dermatology drugs. I think I've seen some shampoo with caffeine. So one compartment model of caffeine, for example, 1 cup of coffee contains 100 milligrams of caffeine in 8 hours simulation. If you can get this curve. But it's interesting that, coffee, you reach some maximal effect, but the minimal effect, which is measured, remains for the coffee like 5-6 hours. In average, but we are individuals, we are different, so somebody needs more coffee, some is missing up with one, or is completely exhausted with one. Somebody prefers black tea. Black tea is, the cup of black tea has almost a half amount of the caffeine. And the green tea also has some caffeine, but for example, in this simulation, you reach that minimal effect only a small portion of the time. So what is the effect? Effect is the study of pharmacodynamics, so what the drug does to the body. And in the Pharmacolibrary, we have several components. First one is linear effect. So based on the concentration, there is some effect. The very few drugs has this linear effect. Most known is ethanol. So if you drink more, you have the more effect, more drug. But the majority of drugs has the, it's called Emax effect, that after you reach some concentration, the effect is quite, slope is steep, but after it reaches some magnitude, then it doesn't make sense to, for example, take another drug because it has no effect anymore. So this is the Emax effect, and some of the drugs also have a sigmoid effect. And so you need also to reach some minimal concentration until it will start to grow to effect. And, so where I have an example of the drug previously presented, it's midazolam and its effect on EEG activity. And based on the data, when the drug is administered, the EEG activity is up. So the patient is sedated in anesthesia, and after some of the activity goes down, then the sedation effect is mitigated, and the patient or the animal can wake up. Here is also example of the pharmacodynamics of the hemodynamics cardiovascular system and pharmacodynamics of digoxin. It is a drug that is used for example, atrial fibrillation or other cardiac diseases. So it is here the hemodynamics modeled in Pharmacolibrary, and you have the pharmacokinetic of digoxin and two effects. One effect affects the compliance of the heart, and the second one affects the heart rate. So when it is simulated, you can get these curves, but what is more important for example, cardiologists is that, and also for students of medicine, that you see the PV diagrams, and after administrating the drug, the PV diagrams go also to the left side, so it has some therapeutic meaning. Last part of this presentation is about pharmacogenomics, so how to systematically approach. We have introduced the records for genotype and for phenotype, and when joined together the genotype are expressed as some mutation in alleles, and it has consequence on the phenotype. So the gene makes some proteins, and for example, the normal alleles here, star 1, has normal consequence on the clearance, but with some mutation, there is intermediate, so a little bit slower clearance in terms of pharmacokinetics. Another phenotype and genotype pair is here. It is in a liver cells, most common cytochrome E450, and again some examples of alleles and metabolizers. This is modeled using the components here. You have the part of pharmacokinetic, and then the influence of the genes and phenotypes. So there is a little bit difference here, but the normal patient with and the patient with some mutation in a gene. But if you have 4 patients with the same condition, you are treating with them with the same drug, you get different results. So, for example, for the normal patient on the right, he never reached the toxic level of the drug, but the other patient, they reach the toxic level because there differs in the genes. So one of the tasks is to set optimal dose of, for example, here the drug lithium carbonate, that the patient after 3-4 days came to the appointment, and they measured this concentration of the drug quite above the maximum level. So it is the task to set the dosage to optimize the drug level. Here, again, visualization for somebody who doesn't know the equations. So it is visualization for the students of medicine, how some of the treatment can affect the toxicity on some cells, and how to find the drug. So here is the Clexane. If you are not, if you are lucky like me and have a broken bone, then you get this Clexane, but it is the name of the drug, the commercial name. You need to find the name of the effective compound, and the name of the effective compound, if it is registered approved drug, it is in the library, and you can find it there. So this is the, and again, workflow with Python and FMI, and connection with the pharmacokinetic database. It's maintained by our colleagues in Heidelberg, Humboldt University in Berlin. So the conclusion, we have the Pharmacolibrary. It's open source. We have it includes all registered drugs there. Some of the pharmacodynamics is also there, but not all. Various effects. In pharmacogenomics, we have only few examples, and we would like to make it also as complete as possible and to contribute to democratize pharmacology. So thank you very much, and I hope that, I'm prepared for some questions.